In this video, however, we will look how we can perform differential expression analysis between conditions of interest in case of multi-sample, multi-condition single-cell RNA-seq data. With this type of data, we mean that we have single-cell RNA-seq data coming from multiple samples and that these samples can come from more than one condition. And in those cases, we are typically interested in finding differential expression within one or more cell populations between the different conditions. If you would perform this analysis, like you would perform it with the pseudo-find markers function, you will pull all the cells together from the different patients and then compare the cells from group 1 versus the cells of group 2. However, this approach treats the cells as experimental units, and this is quite a flawed procedure when you really want to compare samples between different groups. And it has been shown in recent publications that if you do this pooling of all cells of all samples from one condition together, that this might lead to false discoveries because you ignore the variability that you have between different samples. Therefore, this typical approach is flawed and other methods are necessary. In this video, you will learn how to perform a Muscat analysis. Muscat is an R package that allows you to perform differential expression analysis on multi-sample, multi-condition data by treating the samples as experimental units instead of the cells. And hereby it doesn't suffer from the same flaws as the pseudo-find markers function if you would use it on multi-sample, multi-condition data. What we will learn in this video with Muscat is performing differential expression analysis after pseudo-bulking. What does pseudo-bulking mean? It means that we will aggregate all the counts of a gene for all the cells of that cell type, and we do that per sample. So instead of a distribution of a gene expression levels across all cells of a cell type, we will get one singular value per cell type per sample, like shown here on the plot at the bottom right. So we summarize the expression of a cell type of per sample in one value, and then if we do that for all samples, we can later on compare these aggregated expression values of that gene between the samples of the different conditions. And this is called pseudo-bulk, and this type of expression data can be analyzed in the same way like bulk RNA-seq data. So we will use a tool like HR to perform differential expression on this pseudo-bulk expression. In the second part of this movie, we will go over the script that shows you how to perform such a Muscat differential expression analysis. While Muscat itself provides tutorials about how to run such an analysis, it might be quite difficult and also straightforward to adapt their pipeline and visualizations on your own data if you don't have much programming experience or experience with statistics and bulk RNA-seq differential expression analysis in R. Therefore, we will make use of the Muscat wrapper package for this training. This package provides a wrapper function around the functions in Muscat for ease of use and flexibility in tweaking the parameters. And it also provides some more advanced visualizations that are useful in inspecting your multi-sample data before running Muscat, and also some visualizations of the differential expression results afterwards.